We will begin our webinar on behalf of the Madras Medical Mission College of Nursing. It gives me immense pleasure and joy in inviting each and every one of you who are present. I think the participants will slowly come in. We welcome you to the international webinar on neurogenetic approaches to advancing symptom science and psychiatric mental health disorders, which is organized by the Mental Health Department. Before we begin, let's have a word of prayer. Father God, we commit this pleasant evening into the mighty hands of Father. As we join in the webinar and share our knowledge, Lord, and our families, Lord, thank you for the protections thus far which you have given each one of us. Continue to shower your blessing upon each one of us in all that we do, Master, so that we will fulfill the purpose for which you have created each and every one of us. We commit the rest of the session and the technology into thy hands. God, guide, and protect each one of us. In your precious name we pray. Amen. So once again, I welcome each and every one of you. It's a real joy to have you all for accepting our invitation to be with us as we have done a very different approach to the psychiatric nursing in identifying the reasons why our mental health disorders set in. So neurogenetic approach to advancing symptoms is something which is coming up. Probably abroad, it has already come in, and here it will slowly take up a pace. So neurogenetic research has begun in advance our understanding of our genetic variation, which gives rise to individual differences in brain function, which in turn shapes the behavior and risk for psychopathology. More than a century of psychological and psychiatric research has robustly documented that individual differences in personality, mood, cognition, and environmental experiences critically shape the complex human behavior and confer differential susceptibility for psychopathology. The integration of neuroscience, psychology, and psychiatry has shown that variance in the brain circuit structure, and today we are connected together to understand the circuit structure of our mind, connectivity, the resting activity, and the task elicited activation, as well as the peripheral indices of circuit function. So all these have a great impact on our thinking and our thought process. Today we have with us an eminent person who's going to share her knowledge and her expertise. It's none other than Dr. Rosemary Wales, who's with us from the University of North Carolina. Not able to see the screen. Okay. Dr. Rosemary has completed a BSc Nursing in the year 2002 from the Kerala University, India, an MSc Nursing in Psychiatric Mental Health Nurse Practitioner in 2008 from Arizona State University, and Molecular Genetics in 2015, NIH and NINR from the Summer Genetic Institute, PhD Nursing with focus on psychiatric genetics, and doctoral certificate in cognitive neuroscience in 2017 at Duke's University. Postdoctoral research fellowship in neuropsychiatry in 2019 from the University of Pennsylvania, Perelman School of Medicine. Dr. Xavier is an associate professor and has her own research-led lab, genetic lab, a research scientist and a psychiatric nurse practitioner is with us this evening. We are so pleased to have you amidst us to share your knowledge. And she is also an investigator in the biological mechanism of neuropsychiatric symptoms for clinical translation. Equally important interest is in research methodology and analysis guided by the philosophy and principles of open science. Clinically, she has a special interest in psychosis and severely mental ill population. A seasoned clinician with extensive experience working with a severely mental ill and chemically dependent adult population in a variety of psychiatric care settings, including involuntary and voluntary treatment facilities, large community hospitals, state mental health system, and outpatient psychiatric clinic. Dr. Spear is a recipient of multiple awards and honors, including Best Outgoing Student during her BSc Nursing. She was named a Robert Wood Johnson Foundation Future of Nursing Scholar in the year 2014. Recently, she received the American Nurses Credentialing Center for her lab in 2020 and a Certified Nurse Award in the Psychiatric Mental Health Nurse Practitioner category. It's a real joy to have an expert among us and a growing field which we want to learn so many things from you. I welcome you on behalf of the management of the Madras Medical Mission and the faculty of the College of Nursing and the Mental Health Department. So over to you, Madam, for taking over the session through. We love to hear from you and enrich our knowledge. Here is a small e-bouquet which we share to you as a token of our love and appreciation. Kindly accept it. Over to you.
Thank you so much, Dr. Rosalind, Rachel. That was a wonderful introduction, and I'm humbled by what you have described about my work. So let me start screen share. So you can see my slides,
yeah,
right?
yeah, I can see. Yeah.
Okay, all right. So hi, everyone. I just wanted to start by saying that I appreciate you all being here. As was described in the introduction, I'm a research scientist and a psychiatric nurse practitioner. My research interests lie in understanding the etiological basis of severe mental illnesses and to learn how we can leverage this information to improve clinical care. So my lab, X-Lab, it's housed at the University of North Carolina in Chapel Hill in the School of Nursing, and you can find us on the website. So the website information is right here, rosexavier.com. Today, I am going to talk to you about some of my research projects, why this line of research, what we are doing, and how we are doing this. I plan to give you an overview of some genetic methods as well as statistical tools that I use in my research, and I will briefly touch on some methods we are developing in our lab. My goal is to give you an overview of the usefulness of these tools for nursing research, specifically for symptom science and neuropsychiatric conditions. So as you can see here, I've actually sort of highlighted symptom science. So key concept here is symptom science. What is the symptom science? And why should nurses and nursing broadly care about this? So symptom science could it be the culmination of nursing's realization of biology's relevance to the profession? Now, whether we agree on how nursing landed on symptom science, what we can all agree upon is how symptoms are foundational to nursing. We see the person as more than a disease condition. We focus on how illnesses and diseases affect the person as a whole. So we see the person as a multidimensional entity they are. Plainly put, symptom science is understanding symptoms through a disease-agnostic lens. Now, symptom science quantifies subjective symptom experiences. And how does it do it? By measuring the biologic, physiologic, And omic, when I say omic, it encompasses the entire spectrum of omic research. So it could be genomics, metabolomics, proteomics. So anything that encompasses, the overall omic underpinnings of the symptoms and sequelae common to health conditions and their treatments. And this definition was actually proposed by the National Institutes of Nursing Research in their model, what we are going to hear about in the next slide. So the National Institutes of Health, which is the premier research facility in the United States, it's funded by the US government. So the National Institutes of Health Symptom Science model, abbreviated the NIH SSM, is a model developed by the National Institutes of Nursing Research. And what happens is this model offers us some guidance on how to investigate symptoms or how to study symptoms. So you first identify a complex symptom and you phenotypically characterize the symptom using clinical and biological data. You then identify a biomarker relevant to the symptoms using genomics or other investigative tools. Then this knowledge is used for clinical translation or clinical application. For example, maybe you want to identify therapeutic targets, so treatment targets, or you want to screen high-risk populations, or you want to do some sort of test of clinical intervention. So in my lab, I have two major areas of focus: psychiatric genetics and multi-data integration to identify psychiatrically at-risk individuals. So now psychiatric mental health disorders are incredibly complex and burdensome conditions. And even now, we don't know much about the biological mechanisms that lead to these conditions. What happens is the end result is that this limits us from identifying them early and oftentimes treating them effectively. And schizophrenia is actually a classic example. So the poster child of severe mental illness. It's a classic psychiatric disorder where you can see that such burden and disparities are pronounced. My ongoing research projects as such focuses on the genetics of schizophrenia and how we can leverage this knowledge to improve clinical care. Another of my project attempts to identify individuals who are at risk for developing psychotic conditions by using different biological data types that we have. For example, neuroimaging data. So like functional magnetic resonance imaging data, fMRI data, genetic data, clinical data, and so on. But first, I'm going to shift gears here and talk about what makes schizophrenia so complex and hard to study and how we are studying this in my lab. So schizophrenia is a genetically complex and it's a clinically heterogeneous psychiatric disorder. In addition to being a very distressing illness, not just to the person, but the family in many aspects, schizophrenia is also very, very resource intensive. So in the United States, I know we have spent, the numbers are, we spend approximately $156 billion US annually, which includes a combined direct as well as indirect costs. And schizophrenia and related conditions also results in high levels of disability. So a lot of patients receive governmental assistance. So here in the United States, we call it SSDI. Globally, roughly, there are 20 million, so 21 million people who are diagnosed with schizophrenia, and these are the most recent stats from 2016. So it's a very, it's prevalent, and it's actually very, very costly disease. So schizophrenia is a brain disorder, and it's characterized by a constellation of symptoms. So most of these symptoms can be grouped into positive, negative, and cognitive symptoms. So positive symptoms are the dramatic symptoms that is used as a core criteria for the diagnosis of schizophrenia. Movies often portray this. If you have watched the Hollywood movie, The Beautiful Mind, you will see John Nash, who was a Nobel Prize winner, who came up with the game theory. He had schizophrenia. And you would see these positive symptoms portrayed because they are very dramatic. These are hallucinations that could be auditory, visual, or tactile, or delusions or thought problems. Now, negative symptoms include lack of motivation, social withdrawal, and so on. Cognitive symptoms could be executive dysfunction, memory, and other problems with attention. Onset of these symptoms appear to be at different time points. So if we look at the development of a person with schizophrenia, cognitive impairments can be seen as early as 7 years of age. Negative symptoms seem to develop around puberty, and that later comes positive symptoms. What this means is these potentially point to different trajectories of onset and development. Also, clinical outcomes in schizophrenia are also tied to specific symptoms. For example, negative symptoms and cognitive symptoms are most strongly related to social and occupational outcomes. The longer a person stays in active psychosis, the worse outcomes one has. And early identification and treatment is crucial. Now, talking about prevention, that is a completely different story. We are not even close, we are not even there yet. So, I want to talk about symptoms. And this is a very highly complex looking diagram. This is actually what we call as a structural equation model. So rather than overwhelm you, I'm going to talk about what this actually is. So I said the symptom presentations in schizophrenia can be highly complex. But are these symptoms complexly related? So I was interested in studying if there were patterns of associations between these symptoms. And could these patterns of associations offer some clinical value? So this paper is currently published in Schizophrenia Research. And this is the result from one of my studies. So we used a large sample of patients with chronic schizophrenia. The final number of participants or subjects in the final analysis was 1,391. And we used an advanced statistical technique called a statistical structural equation model, or SEM. If you are not familiar with structural equation modeling, just understand that it's an advanced statistical method where you can test for multiple associations concurrently. So for example, when you do regression, you're testing for association between one outcome variable and a few explanatory variables. Here, SEM, you can include a lot of outcome variables in the same model. So the numbers you see here about these lines are what we call as standardized beta coefficients, and the standard errors are in parentheses. So if you don't understand it, I want to redirect your attention to these connected lines. So these dotted lines mean that they are not significant. The connected lines means they are significant. So you can see that in this complex presentation with the symptoms, some of them are related while others are not. Now, additionally, when you examine association of symptoms with clinical outcomes such as treatment response, quality of life, occupational functioning, we know that symptoms have a differential burden on these outcomes. So clinical outcomes are tied to specific clinical presentation. For example, patients with more negative symptoms have significant difficulty functioning socially. Those with cognitive impairments can have occupational and vocational challenges. The first line treatment for schizophrenia, which are medications right now, they mostly target positive symptoms more so than other symptoms such as cognitive or negative symptoms. But the most striking thing for me is that these symptoms have different temporal onset. That is, they first appear, the time they first appear is different, and it differs like by age or in development. So this figure, a complex looking figure, you will see the important neurodevelopmental trajectories that we know are implicated in schizophrenia. So what I want to focus your attention on the genetic and the epigenetic risk. So this red bar here, we know what increases the risk for schizophrenia, and broadly we can categorize them into genetic and environmental risk factors. And why is it important? Because then it makes schizophrenia a very complex disease. Now, there are critical periods during development where the effects of these factors converge to increase the risk of schizophrenia by acting on certain neurodevelopmental pathways or processes. And what happens is that after which there is perturbed or altered neurodevelopment, which results in pathological clinical presentation. For example, we know that from genetic studies that there are many genetic variants which are involved that are associated with schizophrenia, but these are implicated in early neurodevelopment. Now, the effects of these are really, really crucial in the early prenatal period. So here on the x-axis, you will see these are the trimesters. So this is the prenatal period. And then you have the childhood, so until about early adulthood, until about 25 years of age. So the prenatal period is in months, the other is in ages. And on the y-axis, it's basically the percentage of relative contribution to the risk of schizophrenia. So how much do these factors play a role in adding to the risk of schizophrenia? And these are the neurodevelopmental trajectories. These dotted lines, they are not really clear. We have some idea, but it's not really clear. The gray are the neurodevelopmental trajectories, and the genetic and the epigenetic risk, so the genetic variants and the epigenetics or the environmental exposures, act on neurodevelopment at different stages and contribute to the risk, but through entirely different pathways. For example, when you look at adolescents, exposure to cannabis or marijuana, so if you smoke pot, then it can increase the risk of a psychotic disorder, but the pathway it affects is still not clear. Let's take another example. So you see this arching here, that is actually synaptic density. So more connections are being made. So we call it the glutamatergic synaptic density because mostly they are focusing on the glutamatergic receptors. Now, glutamate receptors are one type of receptors in the brain that is really, really implicated in the pathophysiology of schizophrenia. You can see the initial trajectory, which is the second half of the prenatal period. It's not clearly known, but we know that more synapses are growing. And then there's an explosion of these brain, so these synaptic connections in the early childhood period. So like 4 to 5 years of age. After that, the synapses are pruned. So there's called the synaptic pruning. And it's actually, there is a protein called as complement component 4 protein. So the complement component 4 genes are involved in the synthesis of these proteins. They are involved in pruning. And you know what? The strongest genetic association is with this genetic region, so the C4 genes. But what we don't know clearly is how these risk factors contribute to the specific clinical presentation. Why is this important? Because no two patients with schizophrenia look the same. Schizophrenia has a very heterogeneous clinical presentation. And the age of onset also differs by sex. So males usually develop schizophrenia in late teenage, early adulthood, so roughly around 18 years of age. And then you have females where the age of onset is roughly around 25 years of age. Now I want to give you a really quick overview of genetics. So all living organisms have information encoded in the genetic material. So for eukaryotic cells, it is organized into chromosomes. And for us human beings, we have 23 pairs of chromosomes. So 22 autosomes and the sex chromosomes X and Y. Now chromosomes are tightly packed DNA molecules. So if you stretch this out, you will see that DNA is a double helical structure. And it's formed by double-stranded nucleotides, A, T, C, and G. So adenine, thymine, cytosine, and guanine. Now, if there's a change in one nucleotide, so say the C changes to something else, it's called a single nucleotide variation. If a nucleotide in one position differs in individuals, it's called single nucleotide polymorphism, or SNP. Now, the SNP, we pronounce it as SNP. If this change is present in more than 1% of the general population, so it's a population genetic concept, we call that a common variation. if it is present in less than 1% of the population, we call it a rare variation. Now, there are many more types of variations called as structural variations, splice variants. We're not going to go into the details, but say if this T repeats, so then you know it's actually a repeat. So it's a copy number variation. And a cluster of nucleotides can also repeat, and it's still a copy number variation. So with the Human Genome Project, which was completed in 2003, sort of completed, started in 1990. Now, the Human Genome Project was an international effort that helped us determine the entire nucleotide sequence, so the entire human genome structure. So since then, we know a lot more about genetics. So changes in a single gene that can cause disorders, it's called a monogenic disease. So for example, cystic fibrosis, Fragile X, Huntington's disease, and so on. When there are many genes involved, we call it polygenic. And there are environmental factors that play along with genetics, we call it a complex condition. So for example, height. Now, height is a complex phenotype, so it's a complex trait because there are several genetic variants involved, plus there is environmental factors. So if you don't eat well, you're not going to grow so tall. If there's nutritional deficiency, that's what I mean. Similarly, schizophrenia is also a complex disorder because there are many, many genes involved and environmental factors. Trauma, exposure to trauma, cannabis, these things can increase the risk for schizophrenia. So now I want to focus your attention on the first plot here. Now this is called as a Manhattan plot. Now this is actually the result of a genome-wide association study. We are looking for relationships between a phenotype. So here it was schizophrenia, and then you look for what genes are involved. So what genetic regions are involved. In a genome-wide association study, what we're looking for is relationship between Snip, so the single nucleotide polymorphism, with the phenotype, which is schizophrenia. So this is the figure on the left-hand side. So this colorful Manhattan plot is from the largest study to date. So this was published in Nature in 2014. Largest study of schizophrenia, including 36,000 cases and 110,000 controls. So mind you, these are all people of European descent or South Asian descent. So this does not include people of African ancestry as well as South Asian ancestry. And so what you see on the x-axis are the chromosomes. So you can see from 1 all the way to the X chromosomes. And on the y-axis, you see the p-values, which are, to simply put, it's the statistical significance. And for genome-wide association studies, the values or the p-values have to be really, really, really small. So this red line is the statistical significance level, which is 5 into 10 to the power of -8. So normally in research studies, the p-value or the statistical significance, we use 0.05. But for genome-wide association studies, it's really small. So 5 into 10 to the power of -8. So 0.70 and then 8. The reason is we are doing a lot of tests because we have 3 billion, nucleotides, 3 billion bases or 3 billion base pairs. So anyway, so anything above the red line is what we call a statistically significant genetic variant. And we see that 108 regions, genetic regions, are associated with the risk for schizophrenia. So there are a lot of genetic regions. Earlier, you can see that I told you about complement component 4 proteins. So the C4 genes, the strongest association. So this one in chromosome 6, that is actually the C4 gene. So the C4 genetic region. Now, in schizophrenia, rare variations are also implicated. So the graph on the right, so that's the same plot, but we are looking for rare variations. So for example, there is a genetic condition called as 22q deletion syndrome. That is the region on the long arm of the 22 chromosome number 22, a section of the chromosome gets deleted. And what happens is, it can result in an array of clinical problems. So it can, it's called velocardiofacial syndrome earlier, DiGeorge syndrome, an extreme form of that condition is called DiGeorge syndrome. Patients have seizures, patients can have congenital malformations, but someone who has a 22q deletion, the risk for schizophrenia is 30 times higher. So there are rare variations too that can increase the risk for schizophrenia. And the third plot here, I want to talk about, that's actually from my study. We looked at the genetics of symptoms of schizophrenia. And we know that schizophrenia, the overall diagnosis includes a lot of symptoms. So I wanted to see the subset of symptoms were there genetic variants that were implicated. But what we found out was that even for the positive symptoms and negative symptoms, it's still complex. So the genetic architecture, which simply means that all the list of the genetic risk or the variants that are associated with schizophrenia is it looks very complex because there are many of them. Now, I want to point you to, this is the result of an imaging study, so it's a neuroimaging study. They just looked at cortical thickness, which is the thickness of the cortex. Now, I've repeated many times that schizophrenia is a brain disorder. So neuroimaging studies, they provide some evidence for that. And these findings so far has been non-specific. That is why we haven't been able to use these imaging studies to diagnose schizophrenia. But what you see here is, so these are the patients, these are the controls. What you see is the red regions indicate where there is excessive thickening and the blue regions are where there is thinning. In patients with schizophrenia, so change over time, there is excessive thinning. So there's too much cortical loss. We don't know why this cortex thins. We have some ideas, but we don't know exactly why that happens. There are theories that are proposed, but that is one feature that you would see. But is it specific to schizophrenia? You see that, in patients, but you cannot use that to diagnose or to diagnose schizophrenia clinically. Now, so I kept saying about complex phenotypes. So now that you understood that neuropsychiatric phenotypes, so like schizophrenia, is complex. Why is this complex and why is it so hard to study? So if you look at the biological pathway, and I want to just give you a quick summary of what I have said so far. Schizophrenia is a complex, highly polygenic neuropsychiatric disorder. And we know the effects of the genetic and the epigenetic risk factors are seen across the biological pathway, including micro and macro structure of the brain, resulting in significantly heterogeneous clinical presentation. Even when it is this complex, it is essential to understand how these are related, because this information, the genetic variants that are involved, the cells that are involved, the neural circuitry, because all these culminates in a behavioral pattern or a clinical presentation, what we call as schizophrenia. This is important because if we can decode this, then it may help with early identification, may support clinical care, and even prevention. So I can explain this in a slightly different way. It's through the concept of what we call as ontological incommensurability. Simply means, can you measure a concept across different levels? Say the concept of schizophrenia, or what we call schizophrenia. Can you measure it at the genetic level, genetic risk variants? What about the neural circuitry? So patterns of activation in a functional MRI study. And what about behavior? Now, if we measure schizophrenia at these three levels, so the genetic level, the brain level, and the functional or the clinical level, are they going to be the same? Are they measuring the same things? What we do know is psychiatric diagnosis. So what we call schizophrenia is not the same as biology, because you can look at the brain, you can look at the image and say this is schizophrenia. You can look at the genotype and say this is schizophrenia. We also know that psychiatric diagnosis is not the same as symptom phenotype. Say, for example, depression. You see depression in major depression. You see depression in bipolar disorder. You see depression in schizophrenia. So symptom phenotype is not the same as biological phenotype. So for psychiatric disorders, the diagnosis is not the same as biological measurements or symptoms to a lesser extent, and symptoms don't have a one-to-one correspondence with a biological phenotype. For example, there is not a protein that is strongly or correlated to depression. So how do you study this? So now that we agree that psychiatric mental health disorders are incredibly complex, how do we study them? The first step is determining what the question is. So what is the research question that you need to ask? And here I turn to the quote from Jonas Salk. Now Jonas Salk is the person, the Salk polio vaccine. So he said, it comes through asking the right questions because the answer pre-exists. But it's the question that we have to define and discover and to discover and to define. And here I want to introduce you to a very puzzling symptom in schizophrenia. So often patients with schizophrenia are not aware that they are sick or they don't have awareness into symptoms. This is what we call as impaired insight. Now, impaired insight is a multidimensional entity, and it's the awareness of illness. So for example, does the person understand they have an illness? Do the person agree do the person recognize their symptoms? Are they aware that their symptoms are a manifestation of the illness? Do they recognize and understand the need for treatment? Do they recognize the consequences or implications of having this illness? Now, you would think that poor insight, like not knowing you are sick, is bad. But sometimes knowing that you're sick can also tell people that you are different, and that can make you feel depressed, knowing that you're different. So poor insight has bad clinical outcomes. For example, patients don't take medication. So if I'm not sick, nothing in this world is going to make me take medication. So why would you expect your patient to take medication? Poor psychosocial functioning, vocational functioning problems, increased duration of untreated psychosis, increase in relapse, frequent hospitalizations, poor treatment outcomes, increase in voluntary commitments. But good insight can also result in more affective symptoms like depression, anxiety, increase in suicidality, so suicide behavior. And you should know that patients who have a diagnosis of schizophrenia are almost 3 times, like the risk for suicide is actually pretty high, like more than 3 times the risk of the general population. So I wanted to understand if we could biologically understand the basis of impaired insight. And based on a critical evaluation of what we already knew, I came up with a biological pathway model. So this was published in Nursing Research. And what it says is a biological pathway starts with genes encoding for proteins, which are involved in cellular machinery and function. So in the brain, the clusters of neurons and supportive cells, they become neural circuitry, which is the infrastructure for behavior, because the brain determines someone's behavior, perception, and a whole array of functions. In this case, I was interested in impaired insight. Now, impaired insight is a complex symptom, which is multiply determined by biology as well as environmental factors. So this illustration only denotes the genetic components. So there are cellular changes. So for example, if you look at the patients with schizophrenia, if you look at the inferior parietal lobule, you would see that oligodendrocytes, oligodendrocytes are cells that wrap around the axon. It's important for neural conduction, nerve conduction. The number of oligodendrocyte cells are actually fewer in patients with schizophrenia who have impaired insight. And then neural circuitry is influenced, and then you have the behavioral presentation. So when I started my research, no one had actually looked at the genetic basis of insight. So here, I want to revisit the basics again. So earlier I said the schizophrenia was polygenic with many, many regions involved. So it's not a single gene disorder, it's a polygenic disorder. Moreover, the effects of one gene or genetic region is very, very small. So if you have one genetic risk variant, there's no way you're going to get you may not get schizophrenia. Even when you have most of the variants, the risk of schizophrenia is, the genetic risk may be there, but you still may not get the condition. So how do we make sense of this polygenicity? And here, I want to introduce you to the concept of what we call as polygenic risk scores, which is usually applied in situations where there are many, many genes involved or genetic regions involved in a complex condition. So what is a polygenic risk score? It is an estimate of genetic propensity to a trait at the individual level. So to almost oversimplify it, it's just a score. So if you say have 100 genetic variants, you just add up the effects of all the genetic variants, and then you get a score, what we call as the PRS, polygenic risk score. Now it has many uses. You can use PRS to identify genetic susceptibility for the same disorder as the discovery data set. You can look for gene by environment interactions. You can look for gene by gene interactions. You can look for causal relationships, and it may be helpful in identifying patients who are at high risk, or the patients who are at high risk for schizophrenia, so patient stratification. And here are the results from one of my studies, published again in Schizophrenia Research a few years ago, so 3 years ago, 2018. So yeah, 2017, 2018. So I looked at insight phenotypes. So what are the genetic basis of poor insight and good insight? So what you see is for good insight, this was not significant. But for the poor insight, you would see that, so if I group the polygenic risk score into 4 groups, the highest is in the, what we call as quartile. So the highest is in the top quartile, and the lowest, the people who have the lowest polygenic risk score on the bottom quartile, you would see that patients who are in the highest, so patients who has the highest PRS score, so the polygenic risk scores, they had almost a 6 times increased risk for having poor insight. Now, so again, these are just the associations of the different, regions. And just, I want to focus your attention on the bolded ones. So for poor insight, good insight, we didn't find anything. For poor insight, there were 2 regions. So one was in the SOX2-OT gene. The second was an intergenic variant, so we don't know what to make of it. Now, the SOX2-OT gene is very important in neurodevelopment. So as you can see, some of the relevant biological functions are glial state, glial cell fate commitment, forebrain development, negative regulation of neuron differentiation, neuronal stem cell population maintenance. So this is actually very important, and it's associated with poor insight and schizophrenia. So another analysis that we did, we found that it's called a single SNP analysis. Several regions, several risk variants of these SNPs were in the SOX2-OT gene. And here I want to point out something. So when you look at overall genetic studies, findings from these genetic studies, you do want to ask who these studies are conducted on. Most of them are on people of European ancestry, so white people, Americans, Europeans. There are very few studies that are being done on people of color, so African ancestry, Indian population. I know there are very few studies in the Indian population in patients with schizophrenia. So something to keep in mind, we need more studies, we need to understand this. So now here I'm gonna pivot and take a few minutes to talk a little bit about a different method that I use in my lab. And it has strong applications for symptom science. So it's called network theory. Network theory has been applied to psychological phenomena and few mental health conditions, but mostly depression and PTSD. But generally, mental health conditions are conceptualized to arise from an underlying cause. So for example, there's a single underlying cause for depression or schizophrenia, and then you have these symptoms because there's an underlying cause. But network theory challenges this by saying that there isn't a single cause. There may be multiple causes, and so you have to understand the relationship between these symptoms. So for example, a patient with depression can have both problems with sleep and report feeling tired. So if you look at the symptom checklist, you know both are symptoms of depression. But if you don't sleep, well, you're gonna be tired as hell the next day. And whether you're diagnosed with depression or not, you will feel tired if you don't sleep well. So, and this insight is really important to understand mental health disorders. So, what is network analysis? It's a method that can help us study relationship between entities. So a network or a graph is a set of nodes. So these circles are the nodes, and it's connected by edges. And it's represented by the notation G is equal to VE, where G is the graph, V is the vertex or the node, and E is the edge, which is these connections. So in this figure, you see 3 nodes, A, B, and C. The red line means they're positively related. For example, when A goes high, C goes high. A and B, the red lines mean negative relationships. So for example, if A goes high, B might go low. So if you want to look at network analysis to study symptoms, these nodes could be symptoms or these could be patients. So just that's a quick overview of network analysis. And here, this is actually a result of another study. We are getting it ready to be published. So this is from a big sample, what we call as the Philadelphia Neurodevelopmental Cohort, a sample of 5,094 kids, so 11 to 17-year-olds. And these patients were recruited from the greater Philadelphia region. And so this data set had information. So the kids reported symptoms on themselves, and the parents also rated symptoms on the kids. So without going into a lot of details, I included an array of psychiatric symptoms. So PTD is, say, trauma, ADD is ADHD, PHP is phobia. So the entire range of psychiatric mental health disorders and symptoms related to that. And this is just a network that was created from the symptoms that was reported by the kids. And you would see that most of them are positive, just there's only one which is negative. Like the relationship between CDD and psychosis spectrum. So when there is high conduct disorder symptoms, the symptoms of psychosis spectrum is lower. Everything else, it goes high. And I want to show the comparison of the results. So these are the network from the kids, and these are the network, I'm sorry, this is the parents, and these are the kids. They look different. For example, in kids, this relationship exists. For example, there's a relationship between phobia and trauma. In parent report, you don't see that relationship. And also, according to parents, the most important symptom was oppositional defiant disorder symptoms. But according to kids, the most important symptom was depression. So there are differences in how people are reporting symptoms to you. And that is really important to understand. And I want to leave you with one thing. So this is an ongoing project we are trying to make sense of. So can we understand, the biology by combining data? So symptoms, cognition, brain networks, and the genetics. So key aspect here is method development. So we are working on this right now. We should have a few publications soon. So if you are interested in these kind of work, I would encourage you to get in touch or watch our lab space, either on Twitter or the lab website. I think I will stop here, so we'll have a little bit of time for questions. And I want to thank everyone, the funders, as well as the folks who have actually funded my training. So the Robert Wood Johnson Foundation, the Human Brain Project by European Union, Sigma Theta Tau, Western Institute of Nursing, the NIH, NINR, NIH repository. So these are data repositories, the Psychiatric Genomics Consortium, and the Heilbronn Center at the Rockefeller University. So these are just a few people who have funded and supported my work. I think I'm going to stop here so you guys can ask questions. I can address them right now.
Yeah, is there any questions from, as of the chat box? I don't find any. Would anyone like to unmute yourself and ask you questions? That was a real informative session, which I really learned a lot of things. Just looking out if there's any questions. A lot of people who are in the field of psychiatric nursing are there. So participants, would you like to raise any questions, clarify any doubts?
Feel free to get in touch. You can either follow on Twitter or you can follow the lab. My email address, everything is on the lab page. So if you do have questions and you don't feel comfortable asking me right now, feel free to just email me.
Yeah, some of them want me to unmute 1 minute.
All right, let's see if there's Okay, I don't see any questions. So
Yeah, there is no question. 1 minute. They want me to unmute them. Allow participants to unmute. Yeah. It's done. So, participants, would you like to unmute yourself to ask some questions? Please feel
Hi.
free. Yeah.
Hello. Hi,
Yeah,
Dr.
please.
Xavier. This is Prashant.
Yeah, please. We can hear you.
Hello.
Yeah, I can hear
Okay.
you.
Dr. Xavier, this is Prasamsha Haridas. That was a wonderful presentation, and I have a question regarding your Snip variations. So, how early in life can these variants be used to detect the disease condition?
Hi, Prashamsha. So thank you for that question. So the genetic variant, so our genetic structure doesn't change that much. I mean, you're born with your genetic code and it pretty much stays the same, right? So if we can come up with a specific way to calculate the risk accurately. So the challenge right now is we haven't been able to accurately or precisely detect the risk or who will develop this condition. So right now, that's what we are working on. So for example, even when you have the genetic risk variants, having a genetic loading only increases the risk of schizophrenia. So schizophrenia is heritable, which means that if you have the genetic variants, your risk goes up by 80%. So if we can find an adequate tested way that is validated, then we should be able to use it at any time. But the challenge is not in knowing the genetic variants. The challenge is actually finding a way for it to be clinically useful.
Yeah, that's what I'm interested in too. Like, and we use the genetic variants so we can like prepare the patients, or like we can tell them that this is what they need symptom wise. So we
Exactly.
can treat them, like we can have a treatment session for them early in life. That's what my question is too.
Okay, but there are ways to do that. And some of the projects that we have right now, we are actually testing that. We are trying to see how clinically valuable we are. So we have projects ongoing that we are actually testing clinical usefulness of these, knowing these, genetic values. So we just launched a project where we are testing for rare variations to see, does this help, help patients manage clinically? So for example, we are asking physicians and the treatment team to tell us, knowing a person has a genetic variant, or especially a copy number variant, so like 22q deletion, does that help you determine treatment? Like, does it help you choose medications? Does it help you choose referrals? So for example, does this patient need to be referred to a genetic counselor? Does this patient need to be referred to, say, a medical geneticist? So those are the things that we are testing right now.
Excuse me, ma'am. Good evening, ma'am.
Hi.
Hi, I'm Priya. This is for me.
I can hear you. You're breaking up, but please go ahead.
Yes, ma'am. I wish to ask, the schizophrenia is a acute or a chronic? Whether we used to follow the medications for a long time, and I wish to confirm the diagnosis of the condition.
Schizophrenia is a chronic psychotic disorder. So it can have acute, relapses. So for example, someone with schizophrenia, it is considered a chronic psychotic disorder because there is no cure. You can't cure schizophrenia, but patients do recover. So the key thing to remember is, even when patients are in recovery, it's really important for them to follow up with the treatment, because the symptoms can relapse. So the patient can have a psychotic break where they have positive symptoms and they can't, probably put a sentence together. They are hearing voices, seeing things, so the hallucinations and that sort of thing. So it's really, really critical that the patient works with the treatment team. So the physician, the nurse, and the care coordination team, to make sure that they're following through with the treatment.
Yes, ma'am. I have been patient here. So she's had two kids, twins, but she have completely social withdrawal and personality and the maintenance of the buildings and all somewhat lacking. That's why I asked whether she could continue the medicines long time or not.
Well, medications actually don't do much usually for, negative symptoms. So you have to combine. Medications may help with, negative symptoms. What you're talking about, social withdrawal, is a key component of the negative symptom group. They do need social and occupational training, like they need to, there are specific therapies, psychotherapies that can help with that particular aspect of that. So there isn't one treatment that can address, all the array or the constellation of symptoms in schizophrenia. It's mostly about what is, what is available to the patient, and can you combine medications? And the best outcome usually happens when you combine medications with therapies. So for someone with social withdrawal, they need to have some form of psychotherapy. So.
Thank you, ma'am. Thank you very much, ma'am, for the clearance.
You're welcome.
There's one more
Dr.
question.
Devi, I have another question.
Yeah, please go ahead. Go ahead.
So, how feasible is this genetic sequencing, or like for the patient? Is it like the cost-wise?
Well, right now, genetic screening is not done routinely in clinical practice because we don't know if it's going to be helpful or not. So right now, the psychiatric genetics is still in the discovery phase. So what we call as we are identifying the variants, we are looking for what the mechanisms are and that sort of thing. It's not even close to applying to clinical practice, but copy number variants, we are trying to test that. So the first step is to actually do studies where we can test, if testing for these genetic variations would help patients or would help clinical management. So those studies are ongoing right now. Hopefully there is value in doing that because copy number variants are really, really serious. And these are variations of, strong effect sizes. They have been replicated in so many studies. They have other comorbidities, so like medical conditions. So for example, there is this copy number variation called a 16p in the small arm of this chromosome number 16. It can increase the risk for diabetes. It can increase the risk for metabolic syndrome. And that is strongly implicated in schizophrenia. But we don't know what the effects of this particular variation is in patients with schizophrenia. So that's why those sort of studies are currently ongoing. And the technologies, as technologies advances, it becomes cheaper. But right now, the standard for applications of any test, any diagnostic test, be it genetics or anything else, a lab test, to get into clinical is actually very, very high. Validity, sensitivity, all those things are really, really important. So studies are ongoing.
Thank you.
Does it?
Excuse me,
All right.
Hello, good evening, ma'am.
Yes.
Ma'am, I am Mercy. Can you hear me?
Yes, I can.
Yes.
Go ahead, Mercy.
Yes, ma'am, it was a very excellent presentation. I appreciate your interest on the etiological basis, and you have explained us the complex symptoms, the phenotype characterization, and the biometric disorders, the clinical application. Thank you very much, ma'am. I have, as you are interested in the impaired insight, you have talked about the good insight and the poor insight, and you mentioned about impaired insight. Have you found, ma'am, how much does it affect the prognosis of the disease schizophrenia? Can you just say, ma'am, if you have done? And
Yes. So when you look at the prevalence of impaired insight, so roughly 80% of patients with schizophrenia have impaired insight. So which means that not all patients have, this impairment in insight. But impaired insight is often a good prognostic indicator for how the patient is going to do. So as I said, We don't know the exact mechanisms, but if you don't have insight, you're not going to take medication or stick with treatment, and which is a big problem. And that's why you will see patients and their family members saying that, oh, I just wish if he or she would take this medication. When she feels better, she'll stop taking medication, and there goes, she has a relapse. So it's a very strong prognostic indicator for how the patient is going to do.
Thank
Does
you,
that
ma'am.
answer your question? Yeah.
Yes, yes. Thank you, ma'am.
Okay. Okay. All right. Any other questions? All right. I don't hear anyone. So should we just sign off, Dr. Rosalind and Dr. Lata? Should we just sign off? I don't hear anyone else.
Hello, good evening, madam.
Yes.
Yeah,
Hi.
it's a very nice presentation. Yeah, Nanda Kumar here.
Hi, Nandakumar.
Yeah, madam, about the approach of a patient for this kind of genetic testing, whether is it how the patient will be responding there for this kind of genetic testing if you're counseling them for undergoing this genetic testing and all. So because in India, it is the people will not ready to go for this kind of testing and all. So what about the approach there in US? That's
So we have preliminary studies that have actually looked at how patients feel about genetic testing. And
okay.
there is a healthcare system here, Geisinger in Pennsylvania. They did a large study with this. And what they found was patients actually like knowing that it's not their fault, because you don't change your, you don't determine a genetic code. So it's not something that you caused yourself. So a lot
Yeah.
of patients have reported some resolution in their anxiety, feeling that, oh, now, I just got dealt with a bad set of genes, or, so it's not my fault. So patients
Okay.
actually have reported feeling relieved.
Okay.
And the study we have ongoing in my lab, we are also checking for patients, we are also doing this. We are interviewing the patients to understand more about these, like what do they feel? How do they feel relieved? What are the positive aspects? Now, what are the negative aspects? We still need to know, like, do patients see this as a negative thing? So those studies are ongoing in my lab too. We will have results in a couple of years.
Okay. 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 Thank you so much. Thank you.
You're welcome. Yep. Any other question?
Hello.
All right, guys. Yes, go ahead.
Sorry, ma'am.
No worries.
This is Anuradha. I'm a student of Rachel, ma'am. Currently working as faculty of associate professor in psychiatric nursing department at Apollo College of Nursing. Just I want to know a concept of neuroimmunology in psychiatry. Is that anything concept related? Can I understand, ma'am, from you?
I don't do research on neuroimmunology, but immunology is very, very complex. So, and when you combine immunology with brain, which are two extremely complex symptoms, I don't think we have a finger on what that is.
Okay.
But most of the time, there is this immune theory of schizophrenia where I talked about synaptic pruning, right? Like the C4 genes. And the C4, the complement component 4 region. So that's actually an immune response. But that's where we are right now. We just know that there is excessive synaptic pruning in schizophrenia. We know that there is an immune theory of depression where there are, immune responses with interleukins and, those sort of things. That there is excessive inflammation. So you see like very non-specific changes in depression, such as the C-reactive protein. But just because a C-reactive protein is high doesn't mean it much, because it's very, very non-specific. We can't use that in clinical practice. So it's a very, very broad discipline. And that's where I will leave it. It's incredibly complex to study, and you need collaborations and expertise from neuroscience, genetics, immunology, and probably a lot of computational work too.
Okay. Thank you, ma'am. Thank you.
You're welcome. You're welcome.
There's one more question talking about the ethics in conducting such study.
Yes, it's really important. No research study should be devoid of ethical challenges. Is devoid of ethical challenges. That's why we go through IRB approvals, so the institutional review board approvals and all of those things. But more importantly, that's an ongoing challenge, because with the advancements in technology, so the genetic tech, right? The genetic technologies have exploded, right? With like the CRISPR, so the scissors that can edit the genome, those sort of things. But the humanities side, which is the ethics, have not caught up yet. So while we are designing these studies, it's really, really important to combine and think about the ethical, social, legal implications, what we call as ELSI, ethical, legal, and social implications in genomic research. It might look a little bit different from the other side, and there are concepts that are very specific to genetic studies. The NIH, so the National Institutes of Health, there is an institute, the National Human Genome Research Institute. They have incorporated ELSI as a critical part of genetic research. And there are experts. So for example, in my study, I collaborate with a bioethicist here. And in the design phase itself. And the IRB is not going to approve because patients with schizophrenia and psychosis are a vulnerable population. So great question. The discussion needs to happen in the design phase of the study. And when it comes to application, it's going to look very different, because you can get genetic testing like, from Ancestry.com or 23andMe and all of that. What that means, it's going to look very different. So that is a completely different issue, because these companies, biotech companies, are selling these genetic, testing products directly to the public. And within nursing, there's a call for action for us to increase genetic literacy, talk about ethics, because patients are going to ask us, I got the results back. What am I supposed to do? So how are you going to explain the value of that, or the negative sides of it, if we are not educated if we don't understand it properly? So great question, but it's an entire field.
Yeah, thank you for answering that. And I have a question. You had told about the neurodevelopmental trajectories and the interactivity in the environmental factors. As a maternity nurse, I have a concern in relation to the uterine stress and perinatal infections. In your studies, have you found prenatally and intrauterine, what are some of the factors which we have to keep in mind so that we can reduce schizophrenia?
We do know that the intrauterine environment and the stress, the fetus is exposed to within the intrauterine environment is actually an important factor. We don't know the mechanisms though. So one is, if you have the flu, then we've known from epidemiological studies, we know that if you have infections, viral infections such as the flu, the chance of developing, the risk for developing schizophrenia is slightly higher, but more importantly, the intrauterine environment, we don't know the interactions of the intrauterine environment and how that converges with the genetic makeup of the, of the person. So those studies are ongoing. 2 days ago, a study was published in Nature Medicine where they looked at, so remember I talked about 22q deletion syndrome, and that there is an increased 30 times increased risk of schizophrenia if you have a 22q deletion. So right now we can create this mini brains, what we call a cerebral organoid. So this is actually out of a lab in Stanford. They created mini brains. So you can actually create induced stem cells and you can create small organoids. And they compared these tiny blob of cells from patients with like 22q deletion syndrome and normal controls, and they found, changes in the cellular makeup of these. So we have a long way to go, but the tech is advancing, the studies are ongoing, so we are making advancement, though incrementally.
We need to be very careful during the perinatal and intranatal period, which has quite a number of significance upon the manifestation at a later stage. Thank you so much. Now, right, any more questions? Participants, you have an expert before you. Kindly make use of the opportunity to ask questions. If not, you can send your questions. Anything more? Yeah. Thank you so much. There were a lot of participants from all over, from not only from nursing, from paramedical and health sciences, all over Tamil Nadu, many colleges, and initially Dr. Nagarajaya, who was the HOD at the NIMHANS Bangalore, was also with us for some time. I hope sir is there still, I don't know. Right, all the participants, I hope you got enriched with the knowledge which was shared, which is a growing field. We need to understand that mental health disorders has a significant impact upon the family members. So we need to be careful in what we do, identify the cause and set right things so that the future of the family lies in the mental health of a person. So that's more important. Any more questions? Thank you so much for sharing your knowledge with us. I think it's the early hours for you. So thank you for spending time with us. So kind of you, and we will keep in touch so that we can do studies across to find out or identify the causes. So this was I have learned quite a bit today. Neurogenetic approach is something, I haven't touched upon so far. So I have learned quite a bit. Thank you so much for sharing your expertise with us. We appreciate for what you have been doing so far, and we love to hear more from you again. Okay, so if there is no questions, any more? Yeah, right. Over to Dr. Lata, who will give us the vote of thanks. The Vice Principal of the College of Nursing will give the vote of thanks. Lata? Lata, can you hear me? Hello.
Am I audible, ma'am?
You're audible, Lata.
Yes. A warm and a graceful evening to all of you joined us today for this international webinar on neurogenetic approaches to advancing symptom science and psychiatric mental health disorders. The best and the beautiful thing in the world cannot be seen or even touched. They must be felt by heart. Thank you as the one such prayer among them. Firstly, I would like to thank our Lord for his countless blessings for all that we are today and making this event what it was. I'm thankful for the management Madras Medical Mission for rendering us with all possible logistic support towards organizing this event. I must make a remark on the proficient sense of gratefulness to our most respected principal, Dr. Professor Rosalind Rachel, for being the catalyst, mastermind, awakening our passion to reach the pole of success in the best way. Thank you very much, ma'am. I mentioned my deep sense of appreciation, and I'm very much indebted to our chief guest speaker, Dr. Rosemary Xavier, Assistant Professor, Research Scientist, and Psychiatric Nurse Practitioner, the University of North Carolina at Chapel Hill, for the crucial work, sharing with her all her insights, enhancing our understanding to mental health disorders, in spite of all the early morning hours beyond her comfort zones. And we really owe to you a lot, ma'am, for your patience in clarifying the queries of the participants. Acknowledging your effort and connection with us this evening as a token of gratitude, here we are presenting our e-certificate for your deliberation. Thank you again, ma'am, for your commendable talk with us this evening. I'm happy to express my thanks to our teaching and non-teaching faculty for their unflinching support and coordination. I also accord my sincere thanks to our beloved students for their active participation. Thank you, dear students. It has been a tribute to host all the participants this webinar. Without your kind presence, we could not have accomplished what we have dreamt for. So thank you all participants from various institutions for the involvement expressed to us this evening. A big thank you to everyone, one and all. So once again, I thank each and every one of you for being with us this evening. Thank you, one and all.
Thank you. Thank you all for joining. Thank you, Lata.
Thank you, ma'am.
Thank you, all the participants. Dr. Rosemary, thank you so much for being with us. Thank you. Thank you. Thank you all for joining. There are a lot of students from Venkateshwara College of Nursing, Bill Roth College of Nursing. Thank you so much for joining.